In this video, we're going to go over the broad categories of amino acids and then talk about special unique side chains that show up a ton on the MCAT. So without further ado, let's get started. Did you know that every single video has customized Anki cards associated with it that cover the concepts that we discuss in a video? So if you want a way to be able to actively review the material and retain it for the long run, I've got you covered. If you're interested in accessing the Anki cards or even the slide decks within the videos, then check out our Patreon or reach out to me directly for alternative ways to access the Anki cards and the slide deck. Links in the description down below, as well as the links for everything else. So these are the 20 amino acids. You need to memorize these upside down and backwards, including their structure, their 3-letter abbreviations, and their 1-letter codes. So I'd encourage you, once you start studying for the MCAT, start drawing these with the 3-letter abbreviation, 1-letter code, and their names every day until you memorize them upside down and backward. All right, so now let's get started with the broad categories. You have the major classifications include hydrophobic amino acids and polar uncharged amino acids, and finally, polar electrically charged amino acids, and those can either be basic or acidic. So most importantly, don't get the polar uncharged and the polar electrically charged mixed up. The polar uncharged have dipole-dipole interactions or hydrogen bonds with the surrounding environment, and that's why they're polar, but notably they can't carry a positive or negative charge. All right, so starting off with the hydrophobic amino acids, you can pause the video and look at the specific side chains. I don't think there's much benefit in me pointing out the individual side chains for you. But the hydrophobic amino acids come in 2 flavors. You have the aliphatic or alkyl. Alkyl and aliphatic just means hydrocarbon chains. And you have the aromatic hydrophobic amino acids. And this is quite important, as we'll see in a bit, because there are unique things you need to know about the aromatic amino acids. The hydrophobic amino acids overall tend to avoid aqueous environments. So you'll find them at the core of globular proteins, so they'll be embedded deep within those globular proteins, because recall that the surface is going to interact with the aqueous environment, so for example in the cytosol inside the cell, and so therefore the hydrophobic amino acids will not want to be on the surface, and they'll be interacting with one another deep in the core. You can also find them in transmembrane proteins. So recall, for example, the G protein coupled receptor has a component that's embedded within the plasma membrane, and in the plasma membrane, you find hydrophobic fatty acids, and therefore you need hydrophobic amino acids to be interacting with those fatty acids. Finally, they form hydrophobic interactions with one another, and that contributes to the tertiary structure of proteins, and we're going to talk more about the different structures of proteins in future videos. All right, next let's talk about the polar uncharged amino acids. So these amino acids are quite the opposite of the hydrophobic amino acids. They interact with water, with aqueous environments via hydrogen bonding or dipole-dipole interactions. They're found on the surface of globular proteins because, again, that's going to be interacting with the aqueous environment. And there are special cases that we'll discuss in upcoming slides, like special cases, for example, with these that contain hydroxyl groups. But what you'll notice in all of these is that they have some capacity to participate in hydrogen bonding and dipole-dipole interactions. So serine, threonine, and tyrosine have that hydroxyl group, and then asparagine and glutamine have the amide group, and finally cysteine has that thiol group over there. All right, now let's talk about the polar charged amino acids. We're specifically going to focus on the basic ones over here on this slide. So you have arginine, histidine, and lysine. Now, these amino acids are capable of carrying a positive charge because the amino groups in their side chains can carry that positive charge. Now, histidine is unique since its pI value is closer to physiologic pH. So realistically, histidine at physiologic pH will not carry a charge. It'll be in the form of a Zwitter ion or a neutral amino acid, whereas the other two, arginine and lysine, at physiologic pH will carry that positive charge. Now, if you look at the side chain of histidine, you'll understand why that's the case. The pKa of the amino group in histidine has a value of 6.04, so let's say 6. And then the amino group in the backbone has a pKa of about 9. So if you average those, you will get the pi value. 9 plus 6 is 15. And then divide that by 2, 7.5, which is very close to physiologic pH. And that's why at physiologic pH, it will have an overall neutral charge, or a net charge of 0. Now, importantly, these amino acids are often found at the active sites of enzymes, and that's because the active site of enzymes is interacting with substrates and transition states, and sometimes it can be quite helpful to either donate or accept a proton from that transition state to stabilize it. And then you can also find these amino acids in histones. Histones are proteins involved in DNA transcription regulation. We'll talk about this in more depth when we talk about DNA replication and other aspects of DNA. But for now, just know that because DNA has an overall negative charge, histones contain amino acids that can carry a positive charge, such as lysine, arginine, and histidine. And so that positive charge in histone can interact with DNA and regulate its transcription. Next, let's talk about acidic amino acids. So acidic amino acids include both glutamic acid and aspartic acid. Notice that not only do they have that carboxylic acid group in the backbone like every other amino acid, they also contain another carboxylic acid group in their side chain, and that's why they are acidic. Now again, these can also be found at the active site of enzymes because they can donate or accept a hydrogen atom depending on the pH of the local environment. And once they donate that hydrogen atom, they will carry an overall negative charge. So at physiologic pH, unlike the basic amino acids, which tend to be positive, these tend to be negative. All right, now this is very important because whenever I do my live sessions, students tend to mix up glutamic acid and glutamine, or aspartic acid and asparagine, and that's because the three-letter abbreviations, as you can see right here, are very similar. So don't get that mixed up, the three-letter abbreviations. If you see a GLU, that's glutamic acid, and GLN is glutamine. Another way students mix these up is that notice how aspartic acid and asparagine, the only difference here is the carboxylic acid and the amino group in the side chain, but it's still a bit similar because you have that carbonyl group. So sometimes students get that mixed up. And when they take a look at this amide group, it kind of looks like a functional group that can carry a charge because you can see that you have that amine over there. But in reality, amines, when they're part of that amide functional group, so when you have an amine bound to a carbonyl, that's an amide functional group, it's stabilized by resonance and therefore won't be able to accept or donate hydrogen atoms at physiologic pH. All right, so now let's talk about the special amino acid side chains. This is extremely high yield on the MCAT. This shows up all the time, so please pay very close attention. We're gonna start off with the aromatic rings. So the aromatic ring amino acids include phenylalanine, tyrosine, and tryptophan. So notice how they all have these aromatic rings in their structures. Now, what's significant about aromatic rings? If you look at them under UV spectroscopy, you'll see peak absorption at 280 nanometers. So sometimes they'll give you a graph. They'll tell you a protein has a peak at 280 nanometers. Which of the following amino acids is most likely present in abundance in this protein? And it'll be one of the aromatic amino acids. Now, what does it mean to be aromatic? It's not enough to have a ring in your side chain, because proline, for example, as we'll see in a bit, has a ring in its side chain, but it's not aromatic. So you need to satisfy this criteria. It has to be cyclic, it has to be planar, conjugated, and finally, it has to follow Huckel's rule. Huckel's rule states that the number of pi electrons needs to be 4n plus 2, with n being a whole number like 1, 2, 3. So notably, it can't have a decimal place. Now, these little facts, I'll be frank with you, don't necessarily show up on the MCAT. Like, they won't ask you about these, but sometimes they'll describe them in a passage. And I think when they're easy facts to remember, it's quite helpful because you can sometimes skip a whole paragraph if you know these facts, or you can read it very quickly because you already know the fact, okay? So when they're describing this scenario, for example, that tryptophan is involved in the synthesis of serotonin, or tryptophan is also involved in the synthesis of melatonin and B3, sometimes they'll give you like a wordy paragraph describing that. Well, now if you already know that fact, you can literally speed through that paragraph, if not skip it altogether, and save some time when you're answering questions. Same thing with phenylalanine. So notice the difference between phenylalanine and tyrosine is just that hydroxide group over there. So phenylalanine can be converted to tyrosine, and tyrosine can be converted to dopamine with a few intermediates along the way. Again, you don't need to memorize this, but I think it's quite helpful to know because sometimes you'll be able to skip paragraphs or at least a few sentences in a passage and save time. All right, next let's talk about amino acids that contain a hydroxyl group in their side chain. So not only does tyrosine, which we saw in the previous slide, have a hydroxyl group in its side chain, but also serine and threonine. And why is this significant? Because these can undergo post-translational modification. As the name implies, this is modification to amino acids or in proteins after the protein has been translated. Okay, and in this case, we're talking specifically about phosphorylation. So whenever you have an alcohol on the side chain of an amino acid, you can phosphorylate that by kinases, and the purpose for that is to regulate the protein or enzyme's activity, or for signaling purposes, like where it's going to end up within the cell. And specifically, the enzyme that phosphorylates proteins is called a kinase, so don't get it mixed up with phosphatases. Phosphatases dephosphorylate, whereas kinases phosphorylate. More on enzymes in future videos in the playlist. And then we have a little mnemonic over here. Sti can be phosphorylated, so sti over there is the one-letter code for each of these amino acids, so S, T, and Y. All right, next let's move on to proline. Proline disrupts alpha helices, so this is a secondary structure of proteins. We're going to get into that in another video in this playlist, but just know for now that proline introduces kinks into alpha helices and therefore disrupts their structure. It's usually found in beta turns. So beta turns, when you have multiple alpha helices within one protein, sometimes you'll have like beta turns to join those together. You can find proline over there. So if you find it in beta turns, it won't introduce those kinks. And then it also reduces protein flexibility. And the reason that's the case is because proline contains this ring structure in its side chain. But you might object to this and say, didn't we see other amino acids such as tryptophan, tyrosine, and phenylalanine, which also contain rings in their side chain? Well, it's because the ring of proline is quite unique, as you can see in this slide over here. So you'll notice that the ring in proline contains the amino group. So the amino group that's supposed to be in the backbone of proline is embedded within its ring. And because alpha helices, again, we're going to discuss this in a future video, but because alpha helices rely on hydrogen bond formation between the amino groups and the carboxyl groups in the backbones of amino acids, because proline has it embedded within that ring, unlike any other amino acid, it's not going to be able to form those hydrogen bond interactions as well, and that's why it'll form those kinks. So notice the side chain of tyrosine, while it has a ring, that amino group that's supposed to be in the backbone is completely free and not embedded within the ring. Okay, and then you can see I just opened the ring over here. So what I did was I isolated, this is the side chain, and this is the backbone amino group. And you can see once they interact with each other, which is what will happen, it's just, naturally, it's going to form that ring. So once you form that ring, that amino group is embedded within the side chain. All right, moving on to cysteine. Cysteine is quite unique because it contains a thiol group in its side chain. And this thiol group has the capacity to form disulfide bonds with other cysteine molecules. So in order to form a disulfide bond, you need at least 2 cysteine amino acids within a protein. And another thing that's unique about cysteine, although I won't get into too much detail with this because we have another video in the playlist talking about the chirality of amino acids, but it's going to be found in the R absolute configuration when it's in the L classification. So all amino acids that are biologically active are L amino acid classification. And when they're in the L classification, they'll have an S absolute configuration, with the exception of cysteine. When it's the L classification, it'll have an R absolute configuration. But more on the disulfide bond over here. So don't get this mixed up. So the R over here is just referring to an entire cysteine molecule over here with the SH thiol group that's attached to it. And then same thing over here. So this is just another cysteine molecule. The interaction between the sulfur atoms over here is what's going to form the disulfide bond. So essentially what you want to do is oxidize both of these thiol groups. So recall from Gen Chem, we tend to think of oxidation as the loss of electrons. Well, in biochemistry or organic chemistry, you can also think of oxidation as the loss of hydrogen atoms. So you oxidize it, losing these hydrogen atoms, and then the sulfurs will interact with each other and form that disulfide bond. You can see it over here. So this is a cysteine amino acid, and they're interacting with one another via a disulfide bond. Finally, let's talk about glycine. So glycine is achiral, meaning it's not going to have D/L classification nor R/S absolute configuration, and that's because that alpha carbon right there is bound to 2 hydrogen atoms, and when you have 2 of the same atom attached to a carbon atom, it's not going to be chiral. Okay, let's do some knowledge checks and then we can wrap this video up. A researcher analyzes two proteins using UV spectrophotometry and obtains the absorbance spectra shown in the graph. So you can see protein A, the blue line, and protein B is the orange line. Protein A has a peak at 280 nanometers. So which amino acid is most likely present in greater abundance in protein A? Is it V? Well, V is valine. It doesn't contain an aromatic side chain because recall that 280 nanometer peak is going to be associated with aromatic side chains. So V is not the answer. E is glutamic acid, which also doesn't contain that aromatic side chain. W is tryptophan, which contains the aromatic side chain, and therefore W is the correct answer. All right, our next question here. Insulin primarily functions to promote glucose uptake into cells. A researcher treats insulin with beta-mercaptoethanol and observes that the protein loses its biological activity. Which of the following types of bonds was most likely disrupted and by what process? So for the purposes of the MCAT, you should know that insulin contains disulfide bonds. And if you disrupt those bonds, insulin will lose its structure and thus lose its function. Now, even if you didn't know that, you should be familiar with the fact that beta-mercaptoethanol is a reducing agent. And recall how we said the way you form disulfide bonds is by oxidizing them? Well, when you reduce the protein, you do the reverse of that process. You break the disulfide bonds. So here, the most likely bond disrupted was a disulfide bond via reduction. Okay, so our final question here. At a pH of 7, during isoelectric focusing, which of the following peptide fragments would migrate most strongly towards the cathode? So in the case of electrophoresis or isoelectric focusing, the cathode is actually the negative electrode. So it can be a bit tricky over there, but just remember, for electrophoresis, the cathode is the negative electrode. So in other words, what this is asking you is that at physiologic pH, which of the following peptide fragments will have the highest overall positive charge? Well, let's look at them one by one. As you can see in answer choice A, you have lysine, arginine, and histidine, AGL. These are not electrically active. So we're looking specifically on the first four, all positively charged. Although, as we mentioned earlier, histidine is a bit of an exception. So let's just go with +3 for answer choice A. The next answer, you can see you have a bunch of negatively charged ones, so like glutamic acid, aspartic acid, so you can literally just skip this one. All right, now for answer choice C, you have V, R is positively charged, D is negatively charged, F, L, I, A are all uncharged, so the overall charge here is 0. All right, finally answer choice D, just at one glance, you can see that it looks like we have a bunch of positively charged ones here. K, R, all positively charged, so that's +4. But then we also have a bunch of negatively charged ones. So you have the remaining 3 are negatively charged. So the overall charge of this fragment is going to be +1. So the answer here is going to be A has the highest charge, and therefore it'll have the strongest migration towards that negatively charged electrode or the cathode. This is an extremely high yield question. I've seen it a ton on practice exams, so definitely be sure to master this question. All right, I think that concludes our slide deck for today. Remember to memorize these like the back of your hand. So draw them out every day with the 3-letter abbreviations and the 1-letter code. And if you have any difficulties, you know where to reach me. Go ahead and crush that MCAT. If you're interested in accessing the Anki cards or even the slide decks within the videos, then check out our Patreon or reach out to me directly for alternative ways to access the Anki cards and the slide deck. Links in the description down below, as well as the links for everything else.